today, this afternoon. Welcome you all for coming and thank you for being here. Today we'll be hearing the final seminar for Callie Braley. Callie came to us about 4 years ago after a bachelor's degree in agronomy from UNL, and she was a National Needs Fellowship scholar, so she's been working on a number of resistance management issues and things through the course of the last 3 or 4 years. This is kind of the culmination of her efforts today for her final seminar today and her final exam after the seminar. So I guess without any further ado, she will introduce herself a little bit as well when she gets started. So I will turn it over to Callie.
All right, thank you, Dr. Hein. So yeah, it's nice to see some familiar faces here today. So this is definitely gonna make me a little less nervous. So thanks for being here, guys. So as Dr. Hein said, my name is Callie Braley, and today I will be talking about the impact of beneficial organisms in corn agroecosystems today. Maybe if my pointer would work. Okay, here we go. Okay, so just to tell you a little bit about myself, just to warm up. So I grew up on a farm in western Nebraska. For those of you that know Nebraska, it's in the Ogallala area. It's almost exactly 4 hours west from here. My dad is actually a farmer. He grows mostly dryland wheat, some corn and soybeans, and occasionally sorghum. So you could say he's where my interest in plants stemmed from. Okay, at least I got one chuckle. And so this is a picture, actually just a little picture of our farm. Fun fact, this was taken after a tornado had actually been through the area in 2020. And that's my first time I ever saw a tornado. So I was like super excited, as Nebraskans are. But beautiful, I loved growing up there. So as Dr. Hein said, I have a Bachelor of Science in Agronomy from here at UNL. I graduated in December of 2017. When I graduated, I felt like I didn't know as much as I should after 4 years of college, and I definitely had the desire to learn more. And so I founded the Doctor of Plant Health program, and so that's why I'm here today. Through this program, we do 2 internships, and both of my internships have been with CropQuest. CropQuest is an employee-owned company based out of Dodge City, Kansas, and they provide many different agronomic services, such as irrigation scheduling, crop scouting, and precision services. And I've actually been hired with them full-time as a full-time agronomist after I graduate. And so that's pretty exciting that these internships led to that. And so throughout these internships, I looked at many different crops. I looked at a couple fields of sunflowers, some dry beans, alfalfa, sorghum, millet, but mostly what I looked at was corn. So today I will be telling you all about the different beneficials that you can find in corn agroecosystems. And so I will mainly be focusing on conservation biological control. Now, as some of you may know, there are different types of biological control. There's classical or importation, where you typically release an exotic natural enemy to control an exotic pest. There's augmentation, where you rear natural enemies and then release them. And then there's conservation, which is where you preserve natural enemies that are already present. And so later I'll discuss how you can do this, such as reducing pesticide use. So for my document, my first chapter was about what beneficials are present in corn agroecosystems. I talk a little bit about their life cycles and then what pests they help manage. And then in chapter 2, I dive into how do we conserve beneficials. I'll talk about methods to conserve them and then look at a case study of how beneficials can be beneficial when it comes to spider mites. So first, let's talk a little bit about corn in Nebraska. So here in the Cornhusker State, 10.2 million acres were planted to corn in 2020. And Nebraska also ranks 3rd in corn production in the United States behind Iowa and Illinois. Now, it's no surprise there are many different pests of corn. Some are arthropods, some are diseases, and some are weeds. So some of these include western bean cutworm, western corn rootworm, and spider mites, such as 2-spotted spider mites. Some diseases include gray leaf spot, southern rust, and Fusarium ear rot. Finally, as far as weeds, you can find kochia, common water hemp, and horseweed, or some people call it mare's tail, in corn production systems. Now, of course, there are many other pests, but these are some that I've just highlighted because I've had experience with these. So there are many different management options to help control these pests. So for example, that you can use pesticides or cultural practices such as crop rotation. But there's one tool that I feel is a little bit underutilized, which is beneficial organisms. And so during my presentation, I consider an organism beneficial if it helps to reduce the population of a pest. So without further ado, let's get into the beneficials. So I group these by 3 different targets on what they target. So for example, group 1 is beneficials that target arthropod pests. So these range from different beetles to thrips, mites, and parasitoids. Ones that target weeds include ground beetles, other arthropods, and microbes. And finally, beneficials that target plant pathogens include some arthropods and different microbes. So first up, I'll go through this first list of ones that target arthropod pests, and I'll go through each one of these in more depth. So first up, we have lady beetles. Lady beetles are in the order Coleoptera in the family Coccinellidae. So a lot of people, when they think lady beetle, think of the red and black coloration, but not all lady beetles are colored that way. Some are just completely black, for example, like Stethera species. Again, many species in corn, but some include the 7-spotted lady beetle, the multicolored Asian lady beetle, and the 12-spotted or pink lady beetle. Now, these beetles go through complete metamorphosis, meaning an egg, larva, pupa, and adult. You can see the eggs here on this leaf and the larva here. They typically have soft spines or distinctive orange markings on their bodies. So adults and larvae are definitely known for being very predaceous on aphids. However, they also eat other things, such as soft-bodied insects, including mites. For example, Stethera's beetles are known as mite destroyer beetles, just because they're very voracious in eating mites. And you can see this is an adult and a larva right there. However, lady beetles can also prey upon insect eggs, and they can eat pollen. Some can actually complete development on pollen alone. It's important to know that this pollen may reduce predation of pests, though, because it does act as an alternate food source. Some lady beetles are commercially available, but this isn't necessarily recommended because when they're collected, it's often when they aggregate for the winter. And so when they are released, they tend to disperse because that's their instinct. And so their reliability is not that great. However, lady beetles are definitely abundant in cornfields, and so these can be conserved. Next up, we have ground beetles, also in the order Coleoptera in the family Carabidae. These are typically dark colored, shiny, or iridescent. They have ridges on their wing covers, relatively large compared to some beetles, such as a ½ inch to an inch, and their head is smaller than their thorax with thread-like antennae. There are many different species, but a couple that I found throughout my studies include Harpalus pennsylvanicus and Poecilus chalcides. So these also, just like lady beetles, go through complete metamorphosis. So adult and larvae of ground beetles eat many different things, from fly maggots and pupae to rootworms, other beetles, and weed seeds, as I'll mention later. They can consume their body weight in food daily. So due to this and their abundance in agroecosystems, they should be conserved as well. Next up, we have lacewings in the order Neuroptera. There are 2 families I'll discuss: Chrysopidae, which is green lacewings, and Hemorrhobiidae, which are brown lacewings. They have large, lacy wings, protruding eyes, thread-like antennae, and slender abdomens. Again, several species, but one common species that I found was Chrysoperla carnea, as you can see in this photo. These also go through complete metamorphosis. The larvae are pretty distinctive because they have this alligator shape and these sickle-like mandibles that they use to pierce prey. Also, the green lacewings lay their eggs on these stalks, just because to reduce predation from other lacewings because they're cannibalistic, as well as other predators. Brown lacewings will just lay their eggs directly onto vegetation. So all lacewing larvae and brown lacewings are predaceous. Green lacewing adults that are not predaceous will typically eat nectar, pollen, and aphid honeydew. Larvae are very voracious eaters of aphids, so they're known as aphid lions. However, they can also eat mites, thrips, whiteflies, and insect eggs. Some of these are commercially available, but their success has been limited, typically because we don't know a lot of information such as habitat preferences, and so we just don't really know enough to make them successful. So therefore, these should be conserved as well. Moving on to thrips in the order Thysanoptera. There are many different species of thrips. Some are pests and some are beneficials. For example, pest thrips include greenhouse thrips, onion thrips, and western flower thrips. However, beneficial thrips included banded thrips, black hunter thrips, and six-spotted thrips. Thrips go through kind of an intermediate metamorphosis. Some people will say it's incomplete, and some say it's in between, just because they do have an egg, larva, prepupa, pupa, and adult. However, the immatures and the adults look relatively similar, and they have a lot of the same habitat preferences. But for this presentation, I will mostly be focusing on six-spotted thrips, just because they are very important here in Nebraska. So six-spotted thrips are 3 millimeters long. They're pale yellow to whitish in color. They have long hair-like fringes on their wing margins, as you can see here, and they have 3 dark spots on each forewing. So six-spotted thrips typically eat spider mites and their eggs. However, they can also target beneficial mites, which will be the next beneficial I'll talk about. However, as a whole, these mites are very beneficial for several reasons. For example, they can be reproductive and predatory within a wide range of temperatures, from about 65 to 70 degrees Fahrenheit to about 105 degrees Fahrenheit. They can also consume large numbers of prey, about 60 mite eggs or mites a day, and they have a high searching capacity. So these should be conserved as well because they are very beneficial. Next up, we have predatory mites. These are in the subclass Acari, and a lot of predatory mites are in the family Phytoceidae. These are typically larger than spider mites. They're also a little bit differently shaped. Some are a little bit more elongated, as well as they have long legs that are suited for rapid movement. I mean, these guys do move really quickly. There are many different species. There's Phytocelis persimilis, Neocelis californicus, and Neocelis falacis. This last species, Neocelis falacis, is the most important predatory mite here in Nebraska. This one is more teardrop shaped, it's brown or straw colored, and it lacks the pigment spots that are characteristic of pest species. And I'll discuss more of pest species later. These also go through simple metamorphosis. They have an egg, larva, 2 nymphal stages, and then adult. So predatory mites target several things, including phytophagous mites, as you saw in that photo. However, they can also contribute to managing western corn rootworms and wireworms. These studies I looked at showed that the more species that were present, the more impact they can make. So one species of predatory mite alone probably won't make a huge difference, but there's definitely the potential when there are many species together. There are many advantages to utilizing these predatory mites. So for example, they are abundantly available. They have a high reproductive and dispersal rate, good searching ability, and you can find different lifestyles of these, ranging from specialized predators all the way to pollen feeders. So for example, Phytocelis persimilis here is a type 1, so it is a specialized predator of spider mites. Neocelis falacis is a type 2, which means it's a selective predator of spider mites. These are commercially available, but again, not super recommended just because typically it's not cost effective, as well as where spider mites are an issue in hot and dry conditions, those conditions do negatively affect predatory mite survival. However, when we see them, we can definitely conserve them. Next up, we have minute pirate bugs in the order Hemiptera, family Anthochoridae. There are many different genera, but there are some in the genus Aureus, such as the insidious flower bug, as you can see in this photo. They are 2 to 5 millimeters long, black and white, or in the case of the insidious flower bug, it has this gold band across its back. Other species are in the genus Anthochorus, about the same size, 3 to 5 millimeters, but these are black, brown, or purplish. These also go through simple metamorphosis with just eggs, nymphs, and adults. Nymphs and adults of minute pirate bugs are partial to thrips. However, they will eat many different things, including aphids, small caterpillars such as fall armyworm or corn earworm, insect mite, and eggs as well. However, pollen can be an alternate food source, just like we saw with lady beetles. And one study showed that pollen and aphids may reduce predation of other pests. Some of these are commercially available from what I found, though this is not typically recommended or they won't be very effective. However, these are very abundant in corn agroecosystems, and so we can conserve them as well. Next up, we have parasitoids. So parasitoid adults are typically free living, and they will oviposit onto or into a host. Then the immatures will hatch and feed in or on the host, then killing it. unlike parasites that typically do not kill their host. There are two important groups of parasitoids, and the first one is parasitic wasps, Hymenoptera. Some are relatively large, but many are small and unostentatious. For example, in this photo, this parasitoid wasp is just about as big as an aphid. These also go through complete metamorphosis, and there have been several used in successful biological control programs. So for example, I read that there is an ichneumonnid and a braconid wasp that have been used against European corn borer. And it's also important to know that almost all insects are attacked by at least one species of parasitoid wasp. So these guys are pretty abundant. Next up, we have Diptera tachinidae, the family Tachinidae. So tachinid flies mostly resemble houseflies. They have bristled bodies. These also go through complete metamorphosis. And tachinid flies typically go after moth and beetle adults and butterfly and beetle larvae. However, this one in particular, Lidella thompsoni, has been released for control of European corn borer as well. Some parasitoids are commercially available, such as trichogramma wasps, but again, these guys are pretty abundant and it may not be cost effective to release them over large areas. So again, we can conserve these guys. Next up, we have spiders in the order Araneae. So there are many different spiders, as I'm sure you know. They come in different sizes, shapes, colors, but they also have different hunting strategies. For example, some are prey trapping, like this garden spider. They use webs to trap their prey. There are sit and wait ambush hunting types, like crab spiders. And there's active hunters, including jumping spiders and wolf spiders. They target a variety of insects, from beetles to grasshoppers to moths, and they're pretty generalist predators when it comes to what they feed on. These are typically not commercially available, so therefore we can conserve these as well. Next, we have microorganisms. So there are many beneficial groups of microbes, from fungi to bacteria and nematodes. So some examples include Bacillus thuringiensis. I'm sure a lot of people here have heard of it. It's a soil bacterium, and different strains can affect Lepidoptera, Coleoptera, and Diptera. These create actually toxins that are activated when insects eat them, and they create holes in the gut, and so the insects essentially starve. There's also entomopathogenic fungi, such as Metarhizium anisopleae, Bavaria bassiana, which you can see in this photo here, attacking a European corn borer, and Neozygites floridana, which I will discuss later, as this is a fungus that is important in controlling spider mites in certain areas. There's also entomopathogenic nematodes. So for example, there's Steinernema and Heterorhabditis. These have obligate associations with bacteria, and so when nematodes go into an insect, the bacteria will release toxins, and these will decompose host tissues. Nematodes will then feed on the bacteria as well as the decomposed tissues, and so this is what's happening in this photo over here. Some nematodes are commercially available, but they're usually applied as a spray suspension or drenched around the soil around plants. However, there are some nematodes that are ubiquitous in soils, and so these can be conserved as well. Next up, we'll go to the next group, which is beneficials that target weeds. So first we have ground beetles. So I won't go through the life cycle or anything because we already went through that, but there are many species that are weed seed predators. So for example, Harpalus pennsylvanicus is one we saw before. So this one is especially beneficial. A couple interesting studies I looked at studied how different factors influence predation of weed seeds. So for example, this first one on weed seed odors, they tested how ground beetles discriminated between imbibed and unimbibed seeds. They did discriminate between imbibed seeds, and so the researchers concluded that weed seed odors probably had a role to play here. Also, seed depth is very important when it comes to consumption. Both of these studies showed that when weed seeds were on the surface, they were more consumed by ground beetles than at any depth. And so tillage, which I'll talk about later, can definitely change seed depth. And so this could be detrimental to providing food for ground beetles. Therefore, again, they can consume their body weight in food daily, and they're pretty abundant, so we can conserve these guys. Next, we have weevils. And this one, I'll specifically be talking about musk thistle, which you guys may have heard of. It's a weed that was introduced from Western Asia and Europe in the mid-1800s. And while it's not listed as federally noxious, it is invasive, and it's difficult to control over large areas. So there are two weevils that have been used for control. They're in the order Coleoptera crocurlionidae. The first one is the musk thistle flowerhead weevil. This one is 6 millimeters long, and eggs are laid on flower buds. When the larvae hatch, they bore into the flower buds and damage the plant. The next one is the musk thistle rosette weevil. This one's half as big, at 3 millimeters, and eggs are laid into rosettes. When these hatch, again, larvae burrow into the rosettes and damage the plant. Here in Nebraska, the musk thistle flowerhead weevil is the only one that has been well established from what I found. A lot of studies have also focused on just this weevil. However, both of these, all weevils go through complete metamorphosis. So both weevils can contribute to musk thistle management. They target different areas of the plant, again, the flower buds and the rosettes, so they are compatible with each other. However, there is one big drawback that should definitely be considered when using these is weevils attack native Cercium thistles. So here in Nebraska, we have 5 species of native thistles, including Floodman's, Platt's, and wavy leaf. So Lauda et al. Actually showed how Platt thistle can be affected specifically by the flowerhead weevil. In their study, they showed that plants per plot were about 130, and then 5 years later, there were only 20 plants per plot of Platt thistle. So these can definitely decimate Platt thistle populations. So therefore, multiple factors should be considered when utilizing any biological control agents, such as potential non-target effects and host specificity. Next, I'll just discuss a few other examples of arthropods controlling weeds. So for example, there are several insects I found that are for the management of St. John's wort. There's 3 beetles, a gall midge, and a moth that have been used. Next, we have the musk thistle tortoise beetle for musk thistle. For Canada thistle, we have the stem mining weevil and the Canada thistle gallfly. And then we have the bindweed gall mite for field bindweed. So this is field bindweed over here, and I have actually seen quite a bit of this in fields. However, it can be found throughout the United States. This gall mite has been released in only a handful of states though, so its dispersal is not very far reaching. However, when they do get a hold of field bindweed, they can pretty much make it unrecognizable. It just kind of twists and gnarls up the plant, and yeah, it's pretty cool. So again, when we see these guys, we can definitely conserve them, as is the theme of today. Next up, we have microorganisms used for weed control. So fungi are mostly used against weeds, simply because bacteria, nematodes, and other microbes typically lack the host specificity that's needed. Fungi, specifically rust in the Pucciniaceae family, do have several advantages, including host specificity. They also have a high reproduction rate, and they're easily disseminated. Some examples I found are Puccinia cardurorum, which affect musk thistle, and also Puccinia punctiformis, which affects Canada thistle. There have been a few products that are based on microorganisms, but these have not been very successful in the long term. This is typically due to difficult production because a lot of them have stringent requirements for growth, or the market is just too narrow and they just don't make money off of it. So next up, we're to our final group, beneficials that target plant pathogens. So first up, we have arthropods. So some arthropods do act as fungivores, meaning they'll feed on mycelia, fruiting spores, and fruiting bodies and spores. So for example, there's some beetles, dipterans, and mites that do this. Many arthropods also feed on nematodes, such as, again, beetles, flies, and mites. This area of arthropods controlling plant pathogens is definitely understudied, especially in field settings. So their impact is largely unknown, but it's good to know that they are out there. Next, we have fungi. So fungi can directly target other fungi, nematodes, and bacteria. And when fungi parasitize other fungi, they are known as mycoparasites. So for example, there's Pythium and Trichoderma that are mycoparasites. In the United States, many of these agents are not used in field crops, typically because it's probably not cost effective to do it over such a large area. However, they are used in higher value crops, such as ornamentals and vegetables. There's also nematophagous fungi. These have different mechanisms that they use, such as sticky knobs or constricting rings. It's actually pretty cool when I first learned about them. So there's Metarhizium and Nesiopliae. Bavaria bassiana, which can target Meloidogyne incognita, which is a root knot nematode. And you can see this here. And some races can affect corn. And then there's trichoderma, which are typically used in greenhouses. Next, we have bacteria. Bacteria can directly target fungi, nematodes, and other bacteria. So for example, there's Bacillus, which I mentioned earlier. Different strains can be useful for various diseases, as well as there are some that are useful against nematodes. There's Pseudomonas, which can be used for fungi, and then Pasteurella species, which can be used for nematodes. So for example, Pasteurella penetrans can be used against, again, Meloidogyne incognita. Next, we have nematodes and viruses. So again, not a whole lot of information out there, so I just grouped them as one. So nematodes are typically microbial feeders. They will feed on bacteria as well as other nematodes. They are known to reduce plant parasitic nematodes in virtually all soils, but we just don't know how much, so their impact is largely unknown again. Next, we have viruses that can affect bacteria and other viruses. Bacteria, when viruses affect bacteria, they're known as bacteriophages or simply phages. However, their importance for fungi and nematodes, again, is unknown. There are also very few viruses used as biological control agents. And so again, these are out there, but it's just, they need to be studied a little bit more. So that does it for my chapter 1 that I talked about what beneficials are present. I talked about their biology a little bit and what pests they help manage. And so now the question is, how do we conserve these guys? I will talk about methods of conservation and again, go through an example of how beneficials can be useful for spider mites. So we need to ask ourselves, why conserve beneficials? Yes, they help manage pests, but there are also other reasons why we need to conserve beneficials. So for example, there have been a lot of pesticide bans and or restrictive use. These are definitely increasing because there's increased awareness of potential harmful effects of pesticides. For example, the EPA recently announced a ban on the organophosphate insecticide chlorpyrifos because some studies have shown it can be detrimental to children's health. Chemophobia, which is the fear of chemicals, is also pretty widespread in our society based on just my experience. And this influences acceptability just because people definitely want natural food. And I mean, I kind of can't blame them, but it has created a stigma around pesticide use. There's also resistance, which is a really big problem. Overdependence on pesticide has led to a lot of resistance. And there is resistance here in Nebraska. So it hits home. It's in a lot of places. For example, there are a lot of weeds that have shown resistance to glyphosate, including kochia, common water hemp, and horseweed. Western corn wartworm and western bean cutworm have also shown resistance to Bt proteins. And so again, this is a little concerning. And as far as fungicide goes, there's also Cercospora saggina, which is a fungal pathogen that causes frog-eyed leaf spot in soybeans, but it has been confirmed, resistance has been confirmed here in Nebraska to QOI fungicides. There's also been a failure to promote alternatives to pesticides. This one study I looked at claimed that if we had maybe promoted these alternatives a little bit more, we wouldn't have these issues that we have today, or they would be less severe. So therefore, we definitely need to increase awareness about alternatives. And again, one of these is beneficial organisms. So methods of conservation I will talk about include reduced and selective pesticide use, vegetation management, and then I'll touch on a couple of other methods such as tillage. First up, we have reduced and selective pesticide use. Many beneficials are very sensitive to pesticides, and pesticides can be detrimental for their populations. Also, pesticides are often used unnecessarily. They're used prophylactically as insurance against potential pests. So for example, there's neonicotinoids, which are the most widely used class of insecticides worldwide. These can be applied as a foliar spray or as a seed treatment. And I believe in 2011, up to 100% of corn acres, it was between like 80 and 100% were treated with neonicotinoids. So a study I looked at said if there is no pest pressure, then there's no differences in yield. So for example, they looked at corn seed that was treated with neonicotinoids and not treated. And since there's no pest pressure, there was not an increase with the treated seeds. So there was no reason to spend money on it. There was no benefit. Neonicotinoids can also directly harm non-pest organisms that act as alternative prey, such as springtails. And so reducing the use of pesticides helps beneficials and it saves farmers money. If they don't need to apply it, then they can save that money, as there's, again, no point in just using it just in case. If a field has a history of it, then that's a different case. We can also selectively use pesticides or use selective pesticides. So for example, broad spectrum pesticides don't necessarily have to be eliminated, but if there is like a hot spot in a field or just the edge, those areas can be treated alone. We don't have to treat the entire field. Also, we can use selective pesticides. These are definitely easier on beneficials. They're less toxic. Some include indoxacarb and spinosad. And so I definitely want to hit home that pesticides can be a useful tool when used at the right place, time, and the rate. And so they can be useful, but at the same time, farmers can save money if they don't necessarily need to use them, and they should be part of an IPM program and not relied on solely. Next up, we have vegetation management. So establishing vegetation in or surrounding fields provides beneficials with food sources and overwintering sites. So for example, you may have heard of beetle banks. Beetle banks are strips or berms that are planted to native vegetation that promote or enhance populations of beneficials, such as ground beetles, why they're called beetle banks. Filter strips, which are typically used to reduce contaminants from runoff, are also used, and they can harbor beneficials as well. So vegetation can be either complex or simple. Again, as long as there's vegetation there, it has been shown that they can enhance beneficial populations. Also, temporary or permanent vegetation can be used. So for example, beetle banks may be permanent, but cover crops may also provide temporary refuge. So for example, cereal rye is often planted in Nebraska after corn, either with hairy vetch or alone. So again, these can provide temporary resources. So other methods of conservation, just we'll touch on a couple. Using transgenic corn, so for example, BT corn. This is a way for farmers to manage pests. However, it is less toxic to beneficials than insecticides. It has definitely been shown to be. However, again, we have to realize that some insects have shown resistance to BT. There's also, you can adopt reduced tillage practices, as this leaves more residue on the soil surface. This creates more resources, such as overwintering sites for beneficials. It also leaves more weed seeds on the soil surface. So as I mentioned before, tillage can bury weed seeds at different depths, and this reduces consumption of weed seeds, particularly by ground beetles. However, it's important to note that residue can harbor pathogens. So for example, gray leaf spot in this top picture and Goss's wilt are residue-borne pathogens, meaning they hang out in the residue. So this is definitely something to consider. Next, we'll go to the last part of my presentation, which is looking at how to combat spider mites and how beneficials fit in this IPM program. So there are two spider mites that are typically a problem in Nebraska. There's Banks grass mite and two-spotted spider mites. These guys are definitely, they look a little bit similar, but the main difference is their pigment spots. So Banks grass mites have pigment spots running down the length of their body, whereas two-spotted mites have only two spots on like the middle third front half of their body. There are also other differences that are important to note, such as they both produce webbing, but two-spotted typically produces more. Two-spotted also has a much wider host range than Banks grass mites, as Banks grass mites can almost be found exclusively on grasses, whereas two-spotteds are grasses, soybeans, fruit trees, and ornamentals, and other plants. So their timing and their location, Banks grass mites typically show up earlier in the season, start lower in the canopy, and work their way up. Two-spotted spider mites tend to show up later in the season, and they can begin anywhere in the canopy and then expand from there. Also, an important note is their susceptibility to chemicals. So Banks grass mite are still moderately susceptible to several miticides, and two-spotted mites, though, have developed some resistance. So therefore, it's important to know which mite you have in your field. So let's go through their development, just because it's important to know for management. They go through simple metamorphosis, and mites enter cornfields in the spring or summer. So typically they're overwintering in plants around fields, and so they will either crawl into cornfields or be carried by the wind. Once they're there, they will lay eggs on the underside of the leaf and then develop from there. Surrounding vegetation can also influence their movement into cornfields. For example, if wheat is planted in pivot corners bordering a cornfield, when that wheat starts to dry down, the mites will move out of the wheat and into the cornfield. So we can often see mite hotspots start around wheat. All stages can be present at the same time as well, and they typically are. This is a picture I took through a 20 times hand lens, I believe last year. And so you can see a nymph right here, an egg, and these adults. However, these adults are dead, and that's why they look brown, because they had been treated. And many generations can be produced per season. So the first indicators of spider mites are a yellow or whitish stippling on the upper sides of leaves, because when mites feed, they just poke through the plant on the underside and it shows up on the upper side as well. So colonies develop, become larger, and spread across the plant. So for example, in this photo, you can see this is a Banks grass mite colony that's pretty much covering almost the entire leaf. And yeah, they'll definitely move up, but this is one of the leaves on the lower portion of the plant. There are several factors that contribute to spider mite infestations, including host drought stress. Host drought stress is really important because not only when plants are stressed, they just can't tolerate more stress. Host drought stress has been shown to lead to higher leaf temperatures, and higher leaf temperatures help spider mites reproduce quicker, just because that's their biology. And so it's kind of a twofer there. Also, high temperatures, again, help spider mites reproduce quicker, as well as low rainfall and humidity, and a lack of natural enemies to keep them in check. So let's discuss which beneficials are present in corn agroecosystems that can help contribute to their management. So I'll discuss arthropods. So there's phytocoid mites, which can eat up to 15 mites or 25 eggs per day. Mite destroyer beetles, this is a larva here, they can eat up to 75 mites per day. 6-spotted thrips, which I mentioned earlier, can eat up to 60 mites or eggs per day. Minute pirate bugs can eat 30 or more mites per day. And predatory gall midges can eat up to 50 eggs per day. However, these gall midges are not very important in Nebraska. It's just one other beneficial that I found. I think they're more prevalent in like Texas, where the humidity is a little bit higher, but here it's just, the humidity is just not what it needs to be. There's also other beneficials that, for example, lacewings. I've seen lacewing larvae in Banks grass mite colonies. And so this is just a colony of Banks grass mites right here. And so it's important to compare spider mite development to these beneficials development, just to see how much they overlap and how much they can contribute to their management. So spider mites can typically develop in 5 to 20 days. The warmer it is, the quicker they can develop. So under optimum conditions, which is, I believe, at least 80 degrees Fahrenheit, they can reproduce and develop in about 5 to 7 days. As far as beneficials, there's phytocoid mites, which are anywhere from 3 days to 9 days. Mite destroyer beetles, which are 3 weeks to a month. 6-spotted thrips are 10 days. And minute pirate bugs are 3 weeks. And so based on this and comparing them, phytocoid mites and 6-spotted thrips are definitely one of the more beneficial ones for controlling spider mites, just because they can keep up with them a little bit better. They can reproduce just about as quickly as spider mites. So next we have Neozygites that I mentioned. There are a couple species. One important one is Floridana. And this is a fungus that is favored by daily temperatures below 85 degrees Fahrenheit and relative humidity above 90. This does not occur in Western Nebraska. So this is not something you'll typically see out there, but again, in some areas you can see this. And this is actually a female that is infected with this fungus. An interesting study I looked at from Trandom et al. Looked at 2-spotted males and which if they preferred healthy live females or fungus infected females. They actually preferred the fungus infected females to be around. And so the researchers thought this increased the transmission of the fungus. So there are also other tools to combat spider mites because beneficials are very important. They're a very important part of managing spider mites, but sometimes they just can't keep up, whether the conditions are too hot and dry or their life cycles just don't match up. So it's important to know other methods as part of an IPM program. So for example, adequate irrigation can definitely reduce that drought stress and keep leaf temperatures a little bit lower, slow spider mite reproduction, and keep the plants nice and healthy and more tolerable of pests. You can also use drought tolerant hybrids for several of the same reasons, as well as if needed, you can use miticides. Although again, a lot of chemicals are pretty detrimental to spider mite predators. In fact, a lot of spider mite flare-ups can be traced back to when another pest, such as Western bean cutworm, was treated, because it wipes out all the spider mite predators and then they flare up later because there's nothing to keep them in check. So if you will use a miticide, there are some that target eggs and immatures, such as zeal, oberon, and onager. So again, multiple factors have to be considered before deciding to use a miticide, such as how many plants are infested, how big are the colonies, are there mite problems, a history of mite problems in the field, what are forecasted weather temperatures. Agronomists and farmers need to take all this into account when deciding whether to treat with miticides or not. So in conclusion, there are many pests that attack corn, but luckily there are a variety of natural enemies that exist that contribute to their management. Some of these are commercially available, but that doesn't necessarily mean they will be cost effective to use. However, all of these can be conserved through the methods I discussed, such as reduced or selective pesticide use, establishing vegetation in or around surrounding fields, and using less toxic pest management methods like BT hybrids, or using reduced tillage to provide resources to beneficials. So again, beneficials are very useful, as we can see with spider mites, especially when there are a bunch of them together. They can definitely really wipe out spider mite populations if conditions are right. And they are even more effective when used as a part of an integrated pest management program. Again, really trying to keep the plants not stressed with adequate irrigation is definitely critical. And yeah, so definitely good to have them as part of a program and not rely solely on one tool. So with that, I would like to acknowledge my committee, Dr. Gary Hine, Robert Wright, and Tamara Jackson-Zeems, who is on Zoom. And I would also like to thank Callie White. She's been super helpful with a lot of things. Also, DPH alumni and current students have been very helpful throughout my program. Also, my family, my dad, my mom, and my younger sister, as well as my in-laws. They've been super supportive. And then my wonderful husband, who is always supportive, and he's even supporting me right now watching this talk for about like the 10th time. So with that, I will take any questions if you have them.
Questions here? Check to see if we're getting in there.
Callie, conservation is the goal. Some of our common pests you list in Nebraska. How should we be adjusting our economic threshold? Should we? And if so, what around what needs to happen to make a concerted effort towards that?
That's a really good question. I think probably then we need to, I probably needed to look into it a little bit more, but my guess would be we'd probably have to increase the threshold, because we have to account for not just, oh, they've reached a threshold. It's like, are there enemies present there as well? And I do think there are a lot of thresholds that are either really outdated or there's not some for pests. So I definitely think that we should update them and definitely include multiple factors, including beneficial organisms, into that. And that, yeah, that definitely should be a factor. Did that answer your question? Okay.
If anybody has any questions on chat, they can enter it in the chat, or if they can unmute themselves, that would be fine as well.
Sure.
I think you did a really wonderful job giving an overview of the benefits that exist and the rationale for conservation biological control.
Thank you.
What do you think needs to happen to increase the adoption of these conservation practices?
That is an excellent question. And so I think that we definitely need to be having more of these types of conversations. And unfortunately, I've dealt with some farmers like this last summer that the agronomist I worked with was like, well, there's beneficials in here, so we don't necessarily need to spray. And he essentially was like, screw the beneficials, you know, the pest is more important. And so I think we definitely need to make hit home how important some of these guys can be. And some of them are really small, which, I mean, we all like to see things that, you know, are tangible and we can see with our own eyes, such as like herbicides on weeds. I mean, farmers love to see that. So I definitely think agronomists need to be having conversations with growers, as well as in extension, we should definitely, I think, bring this topic up more and more, and definitely hit home on how beneficial they can be.
One question. There's one question on the chat. Basically, you noted a lot of natural enemies in corn. Would you characterize most of them as specialists or generalists?
Good question. I think this is John Riverson. I actually took his class this last semester, so I should know this, right? So based on what I saw, a lot of them are actually seem to be more generalists. There are a few that are specialists, such as certain types of mites, but then there's spiders and minute pirate bugs and ground beetles that definitely can, while they might prefer a certain pest, they definitely seem to be more generalist beneficials. And so, yeah, that would be my answer. They're more generalist than specialized. Thank you for that question.
Go ahead.
A follow-up, would you consider generalists or specialists better targets for conservation and cropping systems, and why? I feel like this is a similar question. Let's see, consider generalists or specialists better targets for conservation. I would probably say, again, generalists as well. This is a little bit tricky because if you're trying to control a certain pest, such as spider mites, then you might want to choose specialized predatory mites or something like that. However, if you use generalist predators, then that means they can feed on multiple things. And so if they're unlike specialized predators, where if they only eat one thing and it runs out, they're probably not going to survive, right? So generalists can definitely stay active, even though that pest that you're targeting might not be present. So definitely pros and cons to both, but I would definitely lean towards generalists.
Any other questions?
Oh, good response. I like that pun. Sorry to be a pest. Thank you, Dr. Riverson.
Well, let's thank Callie one more time. Thank you all for coming. We appreciate your presence both here and online. So thank you very much.
Thank you.